Hello! Welcome.
Hi,
Welcome to YouTube Microbiology Journal Club, where we nerd out big on all things small.
my name is Danny, and in a previous life, I dropped out from my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and in a previous life I got my PhD in microbiology from Imperial College London, where among other things I made flesh-eating bacteria glow in the dark. But I've also worked since then in research integrity, and quite right now I'm working as an editor for an academic journal.
Every week, we meet to talk about microbiology. Today is our deep dive week, where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
Next week will be our News Week, where we survey a bunch of articles and choose an article for the next deep dive. So make sure you subscribe to satisfy your microbiology curiosities.
And you can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you, so please use the comments or tweet us using the hashtag microtwjc hashtag. Um, yeah, so this week we're talking about compliment. Not just normal compliments, like, oh,
Yes.
you look nice today, Danny. No,
The compliments that we carry with ourselves in our blood.
yes. As my professor once said, I compliment with an E, just constantly. Like, your blood's not wishing you a nice day. I'm like, why can't it? Why can't it? But yeah,
Yeah, so to I guess introduce us into this topic, and we mentioned this on the news week as well, that SARS-CoV-2 has been infecting a lot of people around the world, and with all these infected people, we can see the symptoms that come out and notice, like, some symptoms don't appear in everybody, but they do appear in a small population and repeatedly. And one thing that was noticed, this is a table from a paper from like New York City hospitals. They notice an increase in D dimer in the blood. That is a clotting. That's like a marker of like atypical clotting profiles. And you can see like they measured like on average, I guess 438, but like the reference range is 0 to 229. So like this is very elevated in severe COVID cases. And then similarly, if you just like look at ICU, at sort of like the adverse events that come up in the ICU, you get a lot of these thrombotic events, like some sort of strange clotting profile.
yeah. So at the beginning of the outbreak, there was like a glut of papers talking about the endothelial damage, so the damage to the inside of veins and various organs associated with that. And
Yeah, I think it can manifest as like rashes and bruising,
yeah,
kind of like I saw pictures. Some of the papers I think we link in the Zotero like have some cases of people, and you can see like what that looked like in the severe case,
yeah,
right? Very
yeah.
severe case.
It's not pleasant to look at those images, but it is kind of instructive about kind of what is happening in terms of because yeah.
Yeah, yeah. We're not gonna shock anybody today with images of severe COVID-19.
So yeah, so the clotting process is almost a really important thing to think about when we're looking at like organ shutdown or issues like that, people don't often like dig into why the organs are shutting down, what's destroying it. So clotting is
Hmm.
one of those factors that it stops the blood supply to certain areas and can lead to even more severe complications.
Yeah, absolutely. And so, like, I guess we have to understand. We're gonna have to understand something about blood before we continue. Here's a picture of some blood that's being drawn, and then that blood after it's being spun down in a centrifuge. And all that heavy cell stuff goes to the bottom. So that's red blood cells, erythrocytes, and white blood cells, leukocytes, and also platelets. They kind of separate into their own layer. And then the rest of it is plasma,
Mm.
which is just this colorless kind of fluid, which is like packed with proteins.
Yeah, indeed. I mean, if you've been watching the Marvel comics, apparently the vampire Morbius survives on plasma, which because it sounds more sciencey than just surviving blood. But plasma contains all like a lot of the good and plenty that makes the stuff that isn't blood cells. So you've got your white so like complement I believe is in the plasma, right?
Complement is in plasma.
Yep.
Yep, complement, clotting
Antibody
factors.
Yeah, and
Antibodies are in there.
yeah, and I guess sometimes viruses, if you're infected, or other like things that are like not lighter than the white blood cells and the red blood cells, because with centrifuges it's all about
Yes.
mass. So heavier goes down, lighter goes up.
Absolutely, absolutely. Yeah. So in the next picture that we have, maybe you guys have heard of this scientist slash artist, right? But he makes a lot of drawings for the Protein Data Bank. And in this one, you can see how dense plasma is with
Mm.
proteins, right? Like it's easy to sometimes look in biochemistry and in biology, right, and be like, oh, it's liquids, like there's nothing in there. It's just like mostly water. But actually, like you can get a lot of proteins dissolved in water. Um, and that's just, I guess, the artist's depiction based on crystal structures. I guess maybe there's cryo-EM these days in some
Mm.
of these pictures anyway. But from our structural biology knowledge, just how packed that fluid is full of proteins.
Yeah, so I mean, the idea that blood isn't just like a liquid; it's like a complex tissue with its own reactions and the way, like, how it interacts with all the organ systems is a very
Yeah,
important thing. That
it is a tissue.
yeah,
I think people say it's a tissue, and like that's kind of cool when you think about it, right? Like biopsy, like it's so convenient to biopsy blood, right? To take a bit of blood,
yeah,
right, and learn what's going on in your body because it touches all these other things. I mean, we were getting excited in the news week about what was it? Like sequencing blood to look for damaged mRNAs,
yes,
RNA methylation patterns, right? To indicate like damaged organs, right? Like this is the cool thing about blood. It touches everything, so everything kind of gets dissolved into it. And it has its own functions as well to deal with all of those dissolved components.
yeah. Um, and inside blood, I think we've mentioned before, is the complement system. So,
Yes.
I mean, do you want to explain what the complement system is? Because
Yeah.
like,
So this picture, I picked a, it's super complicated, right? Because I want to make sure people know tons of different proteins go into this complement system, but they break them down into three pathways.
yeah,
Usually, they're looking at the classical pathway, they're looking at the alternative pathway, and then they're looking at I don't know what the other one
mannose
is.
binding lectin pathway. So,
Oh yeah, the lectin pathway, right?
like,
And that's just
yeah,
oh,
I
go
was
ahead.
going to go into a little bit of history. Like, so I think when complement was kind of discovered, they assumed it was like kind of helping the adaptive immune system because the original complement pathway it reacts to when an antibody binds its antigen and creates a complex, and then this little C1 like protein comes up and like kind of notices that, and it sets it like kind of it splits up and it causes like
It
some cascade. Yeah,
cascades.
so a cascade is like when one enzyme binds another, and that binds another, and another, and another. So it's like almost like a conga line of enzymes that end up with like
Yes.
passing along a message, and then each part of it, there are certain things that allow them to make decisions so
Mm.
yeah, to make decisions on how to react.
Yeah. Yeah. So I guess they named it complement because it was complementing the antibody part.
Yeah,
But then, right, like it was found out that there's other ways to get into that same conga line, right? To enter that cascade, there wasn't just antibodies that could trigger it. There were also other things. And at the end of the day, that usually leads to some sort of action against the cell that has the complement attached to its
mm.
surface, right? So like if you're attracting complement, like it'll basically it forms, as you can see, the membrane attack complex is one of the outcomes of
Yeah,
complement.
where it
Uh,
punches a hole in a cell, and cells don't like having holes in them, and that causes them to like become drop dead essentially is the point.
yeah, yeah. Uh, I guess what's important maybe to mention here is that like this is like one of the biochemical pathways that that exists in the blood, and like it also touches a bunch of other things, right? We always see um, how antibodies play a role in this. And antibodies are from the adaptive immune system. This is all innate, meaning that
Yeah,
nothing really changes based on previous experiences we've had, right? Everyone has this set of proteins, unless you have some sort of genetic deviation from the norm, right?
mm.
Then you're going to have this full set of complement proteins, and they all will recognize the same things that other people's complement proteins recognize.
Yeah, yeah. So it's not adaptive in any way, but it does take cues from the adaptive immune system, but also takes cues from the innate immune system. Sometimes, like the mannose binding lectins are compounds that are found on bacteria's surface, so that
Hmm.
it can bond and cause a reaction. and the alternative pathway works in a slightly different way. I'm not entirely because some things can so things like cytokines can help promote the complement system. There are various other
Yeah.
like interactions, and sometimes there are things where it'll just act on any surface, and then it'll just wait for the other factors to kind of take. So, for instance, um, it can if it can bind to a target on like the membrane, it can bind to theoretically any membrane, but then there's like this other components that are there to stop it from acting. So if it's got like a certain thing
Mhm.
on the surface that says, "Oh, it's definitely a human cell," like attach, and that'll give a signal saying, "Complement, don't touch this. Where you don't need to do any
Yeah,
action."
yeah. I think that's an important point to bring up, right? Like, because this is like a commonality in the immune system, right? Is like checks and balances,
Mm.
right? Like when your body wields the ability to destroy cells, it has to use it responsibly, right? And like try to only destroy the cells that it needs to. And yeah, and that stuff can be hard coded. It doesn't have to be adaptive, right? It can be like don't recognize yourself. Anyways, let's move on. How does this link to COVID-19? Well, actually, some biopsies, as we were saying, like there's these weird clotting effects that happen. We know something strange is happening in the blood. Also, when you do biopsies of the skin and you look at the vasculature,
Mm.
you can see deposition of complement proteins on the endothelial cells. um,
And that's
yeah,
interesting because it's not necessarily a cell type that we expect SARS-CoV-2 to primarily infect. So there could be
yeah,
something
absolutely.
non-specific happening or something else happening because of the SARS-CoV-2 infection.
Yeah. And I mean, also, I think you wouldn't think that, yeah, widespread deposition of these proteins is also like a danger sign in some ways, right?
Yeah,
Because have these proteins sitting on you, are we target, like, I guess, well, we'll go into the next slide, but I just want to, before we do, I want to say that in B here, they also show co-localization of spike protein with those complement
right.
proteins, at least by
Yeah.
microscopy. Yeah.
So yeah, there is some evidence that there is a linkage between those.
Yeah, there is some sort of evidence of a linkage between those. They even have this like animal study, I guess, as like a linkage between complement and SARS-CoV-2 to try to say that inside of mice, this is from classic SARS,
Yeah.
so not SARS-CoV-2, but perhaps has similar methods of pathogenesis. And they show that in a mouse that is lacking one of the complement proteins, it seems to have get less sick.
Yeah, it loses less weight. There are. I mean, I don't want to go into this, but I know that PenH is a very controversial a very controversial measure, and but that's not part of the paper we're going to
Yeah.
be analyzing. But basically, coming across PenH in 2020 is sort of like coming across like someone reading Reiki off of a mouse. So it's kind of a very surprising thing to
Someone would
see.
be using this metric.
It's something that like kind of when I was coming up in physiology, people were going like, oh yeah, this is something that you should never use because well actually it's a weird sort of irony because they're going, oh you shouldn't use PenH because you don't know what nobody knows what it actually measures. Anyway, here's a p-value, and I'm like, oh okay,
So this is not like the strongest evidence, right, to say that like complement is having an effect, but like it's one of those linkages, right, that brings
yeah.
people down the hypothesis lane.
I mean, I think there definitely evidence that because the weight losses and PenH, it's controversial. People say it measures different things. I mean, that it's almost
Mhm.
like potentially a behavioral measure, which in itself isn't necessarily a bad thing. So it does show that something's happening, but
Yeah.
yeah, it's
So what is PenH? I don't even know. Like to
oh,
me,
and
I saw.
enhanced pause. So the whole body plethysmography, you take this like kind of chamber, you put a mouse or a small animal in it, and then you measure like kind of the changes in pressure in there, and that's supposed to measure like the lung volume. But it's been quite controversial because there are other things that can. So for enhanced pause, it it does a few like wacky things with the equations, but it's very good at like finding differences in certain cases. But it
So it's
doesn't.
like it's a sealed chamber with a mouse, and they're measuring the pressure of that chamber.
Yeah, it's theoretically like kind of a very smart technique. It just there's a lot of data, and you almost have to disentangle. There's various other movement things that you have to think about, and
Gotcha,
also
gotcha. But
like
some of the other, like in D and E, they're using other measures of
yeah,
respiratory. So like they're even though they use PenH, they have some other ones here that may
yeah,
indicate that they still had some sort of respiratory issue
Yeah,
that wasn't bad.
I think that there is enough here to say that there's definitely something respiratory, but I just feel like I have to comment about PenH because, um,
Hey, it's good. I
yeah,
mean, put it out there, right?
it's kind of the well actually kind of bomb on there.
Yeah.
But yeah, apart from that, this still does show because these are mice that were complement deficient, and they
Yeah.
showed that I think that mice that were complement deficient tended to do better than mice that weren't.
Yes.
So,
Yes, they do better.
and that actually
Yeah.
does like sync with a bunch of other studies that like look at the ongoing like kind of systemic problems, and that complement going out of control is a problem in like sepsis and other sort of things. So, I
Yeah, there's good reason, right? That basically, this introduction is trying to build us towards that thought that there's good reason to think that complement is involved in
yeah
something with SARS-CoV-2. So like, these are like the little pegs in the coffin.
yeah
And so in the next slide, I mean, have you heard of this word before? I hadn't. Complementopathy.
um Well, I don't think I've heard of it before, but I feel like I understand exactly what it means because being a biologist, you learn new languages, and if anything with
Yeah.
opathy means it's bad, and complement is okay, and like things like idio, I don't know, pathy, oh, but it's bad, so idiopathy,
Yeah.
that's bad, you know,
And I'm pretty sure that they're also saying here, like, I think if they're saying that it's a similar, like, it has similarities to this atypical hemolytic uremic syndrome. Like, when they say that it's a complementopathy, I think that they were suggesting, like, I just picked this out from this paper. They were making,
mm,
I don't think, it wasn't this paper, I think it was maybe a different paper, but, like, they said it was like, this is the disease that shows some symptom similarities to that severe COVID-19 case.
yeah.
Um,
So this is going to tie into how they measure complement because because this group
mhm.
has worked on atypical hemolytic uremic syndrome, and they've linked it to a lot of complement issues. That
Mhm.
there's like a thrombotic,
And this is
yeah,
and this is specifically the alternative complement pathway
yes,
now that we're looking at, right? So this is one where, and I guess we should maybe briefly describe it, is that um, if your cells are covered with different markers, right, like these C3bs, and if the C3 convertase are activated, they can convert those C3bs into active enzymes, right?
mm,
And then those enzymes are part of this cascade. Well, actually, even C3 having it active is already part of a cascade, but
mm,
once you activate it and it's activating things on the surface, that cell can now perform enzymatic reactions on its surface that recruit the rest of the complement to come and probably destroy it. um, so, but we protect ourselves normally because there's inhibitors, there's factor H, there's factor I. These things
mm,
inhibit that. So our cells don't normally just destroy each other wholesale. So the inability to recruit these important blood factors to the surface of the cell can cause the alternative pathway to activate.
yeah,
Um,
yeah, and
So well, yeah. Anyways, that's the alternative pathway in a nutshell. How it links to atypical hemolytic uremic syndrome is that in those disease states, they aren't able to recruit some of those protective factors, and that inappropriate activation of C3b on endothelial cells, actually, I think
mm,
in that disease, causes this kind of clotting phenomena.
yeah. Um, So I think that brings us like to the background of what the alternative pathway is, how it's kind of been studied in the past, and
Hmm.
now we're gonna and we also like kind of have an idea of how it relates to COVID because we know that from that slide that yeah SARS-CoV-2 and complement they tend to seem to be going together. So um, that brings us to our first figure. Um, so
Oh yeah, I guess maybe I just want to say one more thing from the past disease state
right,
slide is that like that activation on the surface of cells is in atypical hemolytic uremic syndrome, right? In that particular disease, it's a runaway effect,
mm,
right? Because you get a bit of damage once you start to get a bit of damage on the endothelial layer, that's triggering those clotting factors, right? And you're getting more damages. It kind of builds up, and it becomes like that's why it's a disease,
yeah.
right? For people with the deficiency here, like it causes them a lot of problems. And so their hypothesis, right, will be something like maybe the same the pathogenic process that's happening in a hus, right, is something
Mhm.
that somehow that is something that SARS-CoV-2 is generating, right? And they're going to try to investigate that with the tools that they have.
Yes. So we've got our first slide. So what they did was they expressed spike proteins on E. coli, so S1 and S2 separately, and then they added them to. So they did this thing called an MHAM assay, which is kind of a modified ham assay, which essentially you have like a certain cell, and you add human serum to it, and if the complement is activated, that cell will pop and die. So
Yeah.
they've got like a special type of cell called a pig A null TF1 cell, which
Yeah. It's a type of erythrocyte. It's like it's a red blood cell precursor that's missing a certain surface protein.
yeah. So the idea is that when you add that to it, and then you add say either complement or something else to it, or something that will activate the complement. So the idea is that there's complement already in the human serum. So they've got like there's a standard normal human serum that they add that has a complement with them in it, and if they add something else to it, like they can add spike protein. Sometimes they can add some slight acid, and it'll start the complement cascade going off, and then that will deposit on these TF1 cells and kill them, and then you
Yeah.
can measure how much they've died. Um, so this,
And this assay comes from the study of a hus, right? Because
yes,
when you have a person with this disease state, right, and you take their serum out, right, their serum is abnormal, right, for perhaps genetic reasons, and then you mix them with these cells, those cells lyse, right? So like the idea is that because they thought that, maybe something's wrong, right, with there's something inside of SARS-CoV-2, right, that's causing this effect in humans. They're like, let's test it on this cell type, right, that's very sensitive to um, this type of lysis, through complement.
yeah. So so this is an interesting figure because they have. So I'm going to go a little bit off. Well, I'm going to go into the statistics of this paper a
Yeah,
little bit.
let's.
So they use students' t-test for all of their analyses,
Mm.
which is not exactly so. Student t-test is great for looking at a normal population that has like a set number of numbers and comparing just two groups. The
Hmm.
more groups you compare, the more likely you're going to get a false positive result. Um, so in here they're using a t-test for treatment groups, which means that they theoretically are doing many multiple comparisons.
Yeah.
So based on my math, this would be like 15 comparisons. Um, and usually in these sorts of situations, we'd either want to do an analysis of variance, or
Oh,
we usually
that's if they do every pairwise, but I bet what they do is they just do each addition against the control.
yeah. But I
Yeah.
think that it really depends on how they're picking, because it's not necessarily clear that the control is being used as a baseline or whether they're.
Mm,
Uh, but
sure.
so I think the assumption would be that they're going to. So with these comparisons, I think so in these sorts of situations, you use like Bonferroni correction, which is the simplest way of correcting for the things, which is basically taking you take
Multiply.
whatever p-value it is and divide it by the number of comparisons that you're doing it. It's simple
Yeah.
but isn't forgiving. So I mean, if in this case it would probably be about like 0. Well, something around the line of 0.033 or something like that. So uh, again, it's quite difficult to see because they haven't really specified how they've done the tests, and some of these comparisons are implied depending on how they look at them. Or
Yeah.
so, so that this
So
is going to be like
they don't say though, right? They didn't say how they did their t-tests. They
uh,
didn't. Did they say that they did a multiple corrections for it? I don't think
no,
they
they didn't.
did not.
I
Yeah.
believe that they just said, let's see if I can pull this up. All experiments were performed at least 3 times. All data are summarized as mean plus or minus standard deviation
Yeah.
or standard error. The student t-test was used to assess the difference between the unpaired groups. A value of p less than 0.05 was considered statistically significant.
Yeah.
So let's looking at this
And
first
they
figure.
aren't doing every pairwise. They're just it says in the figure legend that it's calculated between each CoV protein treated group and the NHS treated group.
Yeah. So I mean, there is going to be a chance of false positives here, but I don't think that's even the main problem with it, because I feel like doing a t-test or an ANOVA would be the wrong thing, because this is the thing they're trying to do is a dose response. So what you'd
Sure.
want is a regression or
Yeah,
like
I'd like to see
a
this data
correlation.
hitting a trend, right? Of yeah, having more effect the more you put in.
Yeah, because that would really give you a lot more information about like the amount of like how much complement response you're getting per like milligram of spike protein, which would be
Yeah.
useful if you wanted to compare like so they separate S1 and S2 into two different factions. So it'd be really useful if they could compare those two in terms of activation because it actually looks like there is a difference.
Difference. Yes, agreed. But they
It
can't
does,
show that with the data the way that they've done the
yeah,
stat. Yeah, I
yeah.
agree.
I mean, like it doesn't. So I'm trying to look at see because I think this is standard deviation. They don't necessarily mention it, but if it, yeah, so I'm yeah, it's but if it is standard deviation, then that is probably an important difference. But if it's standard error, then it's anybody's guess.
Sure.
But yeah, but it does look like there's a difference at like the higher levels, and I feel like we could have get if the data analysis was done in a slightly different way, we could get more information on that.
Hmm.
Um,
Yeah, I agree with you there. Like if they want to say dose response based on adding this stuff, it would be better to have a statistics that actually showed us dose response rather than just doing pairwise against each dose and the negative control, right?
yeah,
But but it does show us that there is an effect, right?
I mean, yeah, I feel like they're almost missing out on some significance by using these tests. That
Right.
there's a lot of information that we could get out of it that they're not necessarily like we're not necessarily seeing, and it'd be really
Hmm.
nice if we had that.
Totally, totally. Okay, so that's A and B, right? So they're using their test with their heterogeneously expressed, right? Expressed in E. coli versions of S1 and S2. Not those are not the full spike proteins, right? Those are the two pieces
Yeah,
of the
there's
spike
subunits. I
subunit.
mean, if you really want to get nitpicky about it, being like, oh, expressed in E. coli, you might go like, oh, the folding might be different if it's expressed in a bacteria compared to human cells, but the
Yeah.
sequence will be the same. It so it's not
Yeah.
necessarily going to make that big a difference.
Mm-hmm. Mm-hmm. And yeah, and they put them in this MHAM assay, and they see, oh, it's activating the lysis,
Mm,
right? So somehow it makes that normal human serum recognize these cells and bust them open.
yeah, and they
Oh,
test other things as controls. So they test what's it?
nucleic capsid.
Yep, nuclear capsid, which is another part of the SARS-CoV-2, and they also test a different coronavirus spike. So
Yeah.
showing that it's not a generic property of all coronavirus spikes. It's not like the human immune system is already primed to have this idea of okay, well, complement, we're gonna find a spike. It's something else that's happening. But
Something
there's
specific with this particular spike sequence, these specific spike sequences.
yeah, and now they get into some using some complement inhibitors to try
Yeah.
and pass out like what's exactly happening with the complement.
Yeah, so they have a factor D inhibitor that's pharmacologic. So that's like, one of the great things I guess that's come up from like high throughput screens is that like there's a variety of like small molecules out there that inhibit everything. And so they can add a bit of that drug in that doesn't allow factor D to bind. Um, and they see that blocks the ability of S1 and S2 to cause this lysis.
Yeah, so factor D happens quite early on the cascade. So when you think of the conga line, early on the cascade, you're gonna stop all the kind of little branches that'll come off,
Hmm.
and the anti-C5a, that's the antibody to the membrane attack complex. Whether that's the final stage where the complement is starting to do the killing.
Yeah, that's like a little bit later on.
Yeah, that's the end. That's the end. The end of the conga line with a traditional knife to stab people with. Um, that's why I don't like conga lines. I don't go to those parties anymore.
So inhibiting at 2 points, very early in that complement system and very late, both eliminate this phenomena that they're seeing with the S1 and S2 dependent lysis, I guess is what we'll call it.
Yeah, yeah. Again,
Oh,
like
and I highlighted it here in the next slide.
yes, yes, we do highlight. So you've got like two sections we're looking at. So early on the complement cascade, we've got the acting on C3 and the convertase and the enzymes related to that. And late
Hmm.
stage is the enzymes that are creating the membrane attack complex.
Yeah. Okay, so they've shown us a little bit of evidence. Again, this is just this assay is really just to show like they've repurposed something, right? This
Mm.
assay is showing us like is supposed to show lysis when atypical hemolytic uremic serum is applied onto the cells. So, but like because they've made this argument that like oh it could be really similar to SARS-CoV-2, then like this is what's giving them, I guess, their initial boost of confidence to go mechanistically now and try to dissect what complement proteins are required to make this happen.
Yeah, and so they use flow cytometry, which is I think one of our favorite ways of measuring cells. So they use like various antibodies to look at the deposition of C5a, C3c, and C4d.
Hmm.
So these are all different parts of the. So C5, we've talked about membrane attack complex. C3c, that's like almost a decision-making protein, and C4d, I can't, I mean, remember what this was because I think I wrote it down in my notes somewhere. Um,
I think C4D is also in the decision making of one of the.
oh yeah, that's with the mannose binding lectin and the kind of the traditional pathways. I feel so. C3c is more of the alternative pathways.
Yep.
C4d is activated by antibodies and this mannose binding lectin protein.
Yep. And C5 is that end again,
Yeah.
right? That's directly leading to the effect. And like so this is just maybe you don't trust the lysis. You think lysis could happen for any reason, right? Like there could be other things
Yeah.
going into that. Flow cytometry is trying to remove that part of the doubt, right? And say, no, we're seeing deposition of those complement proteins on the cells.
Yeah.
um, so a little bit more direct way of looking at it. And so I didn't really get this, but I think that this is the case. I don't understand complement fully here, but I'm pretty sure they have different buffers because different complement proteins need different cofactors. That's what I
Yeah.
was getting from
That,
reading the
that's right.
composition of these buffers.
So I think for some complement factors they require some ions to be part of the process. So calcium, magnesium. Uh, so um, so they use this GVB kind of buffer, which you can either add calcium and magnesium to turn it to GV++, which will activate the classical and lectin pathways. But without that, then only the alternative pathway will happen. So
Right.
it's just like making sure it's got all the ingredients because, as you said, complement is a complicated thing and it does require lots of different factors in order to work.
Yep. Yeah, so they're trying to eliminate they're again, they have this idea that it could be alternative pathway because that's what's wrong in atypical hemolytic uremic syndrome, right? But they want to show more down that path. And in this way, they are doing it by removing those critical components, ions
Mm.
that are needed to go down the other paths, and they essentially redo their test. Right? That's all that this is. But this time they're looking at the deposition of that end molecule C5b on those cells.
So yeah, I mean, I hate to harp on about the statistical analysis, but this is really unclear what they did for this because t-tests deal with column data, and where so I mean I don't think that they went as far as to measure compare each individual like kind of point on the
Yeah,
line because they only put the stars right at the end. So that indicates that they've done something maybe to the whole lines, but it's
that's weird.
really not.
That's not how you would do t-test, right? Not for lines.
Yeah, it's very very unusual. Um, I mean, yeah. So
I
I'm
kind of made
fully
the assumption
baffled.
that they just did the final time point. That was my thought, is that, but they don't really say, I don't think.
I mean, yeah, I feel like if they did the final time point, then those two graphs, the significance, like kind of stars, would be the opposite, because you can see, looking at the looking at what I'm assuming are standard deviations. I mean, if it's standard errors, then as I've said before, you don't know. But standard deviations, then that overlap would mean that that'd be kind of not significant. So it's complicated to interpret these without knowing like what kind. So at this point, I'm just taking all of my thoughts about statistical significance and kind of putting them in a box somewhere because I don't think that any significance stars we see on this will actually be helpful. I think at some points it might actually make us uh, doubt their analysis a lot more than we should. Like,
Sure, absolutely. Well, I mean, I think maybe that's an important point to bring up, right? Like, statistical significance is not the be all and end all of understanding data, right? Like,
of
we can
course,
also look at the difference between things, right? As like, right? Like, and people don't do this type of statistical analysis that much, right? But you can look at the difference between the absolute, like, difference between the 2 points, and then calculate confidence intervals to see if those confidence intervals will overlap.
yeah, that's a kind of a very rough way of. I mean, I guess statistics give us an idea of the certainty to what we believe to the data to be, and I don't
Hmm.
think that's this all is all very kind of very preliminary,
Hmm.
and the thing is, when you've got like inappropriate statistics used on something, then trying to judge it based on those inappropriate statistics is actually kind of unfair, and you have to like take it out of context because the data is still the data, but it just we need to take away all our ideas of like how certain we are about this data, and
Yep.
just try and go with our gut. I mean, it would have been helpful if we had like data points shown properly, uh, and let me because I need to double check because yeah, it's really unclear whether these are standard error or standard deviations. Um,
Yes, I did not see that information. I also kind of, when you first brought it up, I'm like, oh, maybe they say it somewhere. I guess I could search the paper to look for either or.
yeah, just giving a quick check of that. I mean, I've only got the paper copy,
Oh
so my
yeah,
search
they use.
algorithms.
Oh no, they say standard deviation or standard error.
So they use yeah, that's kind of the various things that make me kind of very much scratch my head and go, okay, but at least make sure you're using the same one in each graph, right? I mean, I'm hoping that they do that, otherwise, because you can,
I mean, this is interesting, right? Because this kind of this was one of my pet peeves, I think, going through grad school sometimes, is that people will use their like favorite graphing software. It was
yep,
GraphPad in the lab that I was in, and they would just let it say whatever it said, right? If it
yeah,
outputted the stars and like it looked like a graph that you would see in a paper, they would use it sometimes. Of course, like the best supervisors would be questioning those things, right? And saying
yeah.
like, I don't know if that's appropriate, but stuff slips through, and then
Oh,
sometimes people don't care that much.
yeah, I mean, my old supervisor isn't the kind of person to shout at some people, but she was definitely the kind of person to like raise an eyebrow when if I was trying to pass off standard error as standard deviation, or like kind of
Yeah,
was very clear that they aren't the same, and that one of
yeah,
them is very informative, and one of them is makes your data look nice. So
yes, yeah.
yeah, this is and other things that kind of make me kind of raise my eyebrows is saying like, oh, at least three. So they use the term like what was it? Repeated at least 3 times. So that means that sometimes they repeated more, and that means that some time points are giving us more information about the overall variance than others. And without
Hmm.
knowing what those are, it's quite hard to because that
Yeah,
means you might have to
yeah,
weight some like standard errors and kind of some distributions a bit higher than others, and
yeah.
it's just
We're railing against the way the data is being shown to us, right? Like I think that
yes,
that's the theme. That's the theme of these rants. I think is that
yeah,
like the data being shown to us is not giving us a good idea of what is going on, right? Like it might look kind of nice, like in terms of like, oh wow, like lots of stars means the difference is there, right? Look at these lines, it means stuff is happening over the time that they're sampling. But actually, I think we may agree that maybe if they showed us the scatter plot of these things, we'd see all the dots. We'd know how many times things were repeated in each one.
yeah.
If they told us specifically what these error bars were, right, and then how that t-test was done, because yeah, it obviously it wasn't done on the full line to get this
Mm.
amount of significance all the way through.
So
Yeah.
yeah, I mean it's weird because I think there is a kind of fear about doing that as if your data will disappear, but I think it actually strengthens it some because I remember one of the first videos I did was where they didn't do these analyses and they didn't find they said oh it's probably not significant and then I did the analysis myself because they put all the data out found they did find something significant and it's for so it's kind of frustrating when you have people have all the data there and you're like oh you could give me a great answer here and they're like
Yep.
yep job done it's fine and I'm like I'm like a
Yeah.
little bit like I want more.
Yeah, because I mean, statistics knowledge updates as well, right? Like as
Mm,
different cohorts of graduate students go through, we were just speaking like we're not super Bayesian statistics oriented,
no.
right? But like hopefully as new graduate students go through the system, they will be, right? And they may demand different types of statistics done on the data. Best to just show all the data,
Yeah.
right? And then let people like make their own decisions. And of course, you're guiding people down a story. You want to show your analysis as well, why you decided to then continue down a certain line of reasoning. But yeah, there's utility to showing the data not as just an average dot with with bars, indeterminate bars around them.
Yeah. So hopefully, like, so again, I'm gonna try and put the
Okay,
statistics
let's move
back in
on.
the
Let's move
box.
on.
Back in the
Yeah, yeah,
box.
back in
Just
the box. And again, focusing on what we think this figure
yeah.
itself is revealing to us, right? And there is a difference between A and B, right? I think that that's kind of the important point. There
Yes.
is this like seeming difference between A and B. You know, going, oh my god, I'm going to reopen the box, but like because like you would want to see you would actually want to see the statistics between A and B, right? What's
Yes.
like, right? Like I'd actually want to see, does the S1 line change significantly between, right, using the all pathway buffer and the APC specific buffer,
Yeah.
right? But we're not given that comparison. We're given them in two separate graphs, and we're just like eyeballing it to be like, okay, it doesn't really change, but yeah, it seems like it changes for the spike protein. Who knows why, right? And they haven't built the story up to go down that path. Okay, but let's move on. I think, right?
Yeah.
We've
Let's
talked
move
about.
on.
Okay.
Okay. So figure S1. Yeah, this is like heat killed spike. So they took spike and they subjected to heat, and heat will change the structure of the spike protein and change so it won't be able. So usually it won't be functional. So and
Yep.
they kind of find that that decreases the amount it activates complement.
Yeah.
Uh,
I think that's good. It's a good control. It's like they're shoring up a little bit. Probably the heat treat. Like I'm guessing that it doesn't obliterate it, right? When you
yeah.
heat in earlier tests, I hate to, but if you remember, like from the
Mm,
very beginning, they actually use heat treated not human serum
yeah.
as a negative baseline, right? Because once you treat that human serum, it doesn't do anything anymore.
Yeah.
Here they heat treated like this little fragment, right, to try to break it. It breaks it. You still see an effect. That's probably because it can refold. It's only
Yeah.
a tiny object. It maybe can find a way.
Yeah.
So yeah, anyways, just some insight, I guess, into
Yeah.
the biochemistry of S1 and S2.
Yeah, whatever site might not be destroyed completely. I mean, it would have been nice to. Oh, I hate to do this again, but it would be nice to have a baseline like control on these as well, where it's
Uh-huh.
like the N protein or the OC43.
Yeah.
That
To
would have
see the
been nice.
comparison. Agreed. Agreed.
Yeah,
Agreed.
yeah. I
Okay.
feel so bad because people put a lot of work into this. I don't want to like poo-poo them because I've been in this position where I put up
Yeah.
my work in front of a supervisor and they're like, "This is terrible." I'm like, "No, no, it's there's some good here. It does show something, but
Yeah.
it's
Yeah. Yeah. We're not debating the basic things that it's
yeah."
showing, but it could show us so much more given that they already did a lot of this work, right? They had these tools at their fingertips.
Mm.
Um, okay.
But we're
Uh,
not saying it could be better, is what we're saying.
yeah.
Uh,
Figure three ACE. So they go back, they're still using their flow cytometry. And now they're going to observe when they add in that factor D inhibitor or the anti C5 antibody, do they see less deposition? And they do.
yeah, they do. So I mean that's kind of what you hope for basically. So that kind of demonstrates that C5 is having an effect. Um,
Yes. Yeah. I mean, to me, the antibody is great, but like again, to show something well in science is to show it from different angles,
yes,
right? I think that this is that other angle, not just lysis, but the actual observation of the protein aggregating on the surface. And here you can see that it represents a flow plot. We've talked about flow before, right?
mm,
But you see that shift in fluorescence, right? So just to remind people that the S1, right, how it has that broader peak, right? It means that in the population of cells, a bunch of them are picking up this S1 and they're glowing more in the S1 band.
yeah, and that brings us to Figure 3D. So uh, now we're looking at blocking C5a, right? And
Yeah,
no,
the same thing, blocking C5a and factor D, but looking at the C3 deposition
yeah.
on the surface. Yeah.
So C3 won't be changed by this at all because C3, as we've recalled, is right at the beginning of the conga line, and we're cutting things off right at the end. So C3 won't will still behave in the same way, and that's kind of what we're seeing as well.
Yeah, to me, actually, this is one of the most important figures of the paper because C3 is that part of the alternative pathway, right?
Mm.
That based on their reasoning from the introduction, right? Their reasoning that it's this alternative thing, right, that is causing the issue. So yeah, I think that this is a very important figure. Yeah.
Yeah, I think that really helps. So just to highlight what we were talking about, so oh no, we're looking at C4 now, which is a
Mhm.
part of a different part of the pathway. So that's more towards the mannose binding lectin and the traditional complement side of the pathway. Seeing how that gets affected by S1,
Which is not very much.
not very much at all. So they've got Shiga toxin as a control. So Shiga is like has those kind of mannose those lectins that get bound by the MBL. So that gets get quite activated by. So that's our positive control and a negative control, which I quite like that they have that. I like.
No, I agree.
Yeah.
And after seeing this data, would you feel comfortable saying that there is some sort of alternative pathway phenomena at play here, right? That is being triggered by S1 and S2?
I mean, I don't want to be pinned down on saying that
in,
completely,
in
but
vitro, in vitro, in
in
vitro.
vitro in this situation
Yeah, in
with
vitro.
the way this data's been represented and I'm I, look, I feel like there's enough to say that there's something there, but I'm not quite really willing to be pinned down on saying that this is like kind of going to happen the same in everybody's petri dishes. Um,
Okay.
so
Oh
that
yeah, that is a very important point. Yeah, like the lack of like for you seeing these statistics presented in like sort of a not satisfactory way, right? Makes you think, what if I took this up in my own lab, right, and did these
yeah.
experiments again? Would I see the
So
same effect?
this would be less because sometimes you see a paper and you can go, okay, well, based on that, I can take the next step. This is where I go, okay, I want a preliminary to repeat some of these experiments and then see what I find there. Even use these to power like a preliminary. So when you're doing an experiment and you're planning on it, this will be giving you an idea of what to be looking out for, and then use that to power something a bit more detailed and a bit more in depth, and then
Sure.
you'd come back to. So it's
Yeah,
so
I'm all for that. That sounds like a very rigorous approach to take, right? But looking
yeah,
at what you've seen,
that's
based on
what
what we've seen.
that's basically what my supervisor would make me do because and like sometimes because taking the next step you kind of can fall apart because I know like some of the experiments I attempted that failed during my undergrad were always due to when I look at paper and go, oh, if this happens then I'll just do the next step without doing that preliminary to confirm what they've already found and whether it will work in my petri dish or not. So it's evidence, I think that, but you just have to take it in context and no.
Mhm.
So it's um
Yeah, I think to me the most important thing, I think I would feel more confident if they showed me the significance between the inhibitor and the S1, S2 addition in that
mm,
assay. If that's the only data that they can show, right, and they can show like a clear difference, right, in multiple replicates, then to me that's actually the critical piece of information for me to say, all right, something is going on. So between factor D and S1, like this is like from figure 1EF or
mm,
whatever, I feel like that would make me feel good. Oh, and then do it again with the weird buffer switch because that's like an
yeah.
alternate way, right? And then do it again with the flow cytometry. And like I think that would be like, yeah, something's going on. Like the
Yeah,
fact that when you remove this part, this very specific part, right? Like pharmacologic inhibition is pretty specific, right? Removing factor D from the equation shows us that that breaks down the ability of S1, right, to enter the human serum and perform this effect. That would make me feel good about the in vitro. Okay, but
yeah.
let's keep going because they
I
keep
mean,
doing in vitro stuff.
yeah. I mean, we
Yeah.
weren't there though, so we don't know whether those machines were broken down on the day or whether they had. And everyone knows that with SARS, everything's rushed in double time as well. So
Oh
sometimes
yeah.
there are some shortcuts taken, and we're not there in the lab, so we don't know what the situation was. So we don't want to necessarily judge. I mean, we just have
Yeah.
to
Well, I
take
mean,
what
I
evidence
guess
they could find. Yeah,
yeah. I mean, everyone's different, right? And what data that they think is best, right? Like they're arguing to us, right? Like this is the
yeah.
argument, right? And I'm just saying, like, oh, I think I would see this as a very succinct argument to where they've brought us so far, where they
Mm,
brought us so
yeah.
far.
So next we're going to be looking at the C3 end of the pathway again, looking at figure
4A.
4E. Yes. So yeah, with
Yeah.
PB.
Yeah,
which is a
kind
product
of
of the
activating the C3, right, on
hmm,
the surface, or the C3b, I guess, right, is yeah, the ability to split up to I guess there's some sort of proteolytic processing of BB that
yes.
happens, and it gets released into the serum. You can read these levels as preempt, like the beginning of activating those C3bs on the surface.
Yeah, and this
Hmm.
does seem like there is I mean, more. There just seem to be a more clear effect here.
But this is what I'm saying. Like, there's some clarity in just these like
Yeah,
single comparisons because their
yeah,
tool is t-tests, right?
yeah,
I think it brings a lot of clarity when they show data like this.
yeah. I think that there does look to be like a big difference in S1 with the the absence of. So what were they comparing against? The presence of cells and the absence of cells,
Yeah.
and the deposition of and
Yeah,
this the
like
amount of.
yeah, but what I'm saying is even in just NHS versus S1 in the presence of cells, right? That's the old assay. They've done that
Hmm,
before, right? They're
yeah,
just showing us another metric, right? Now serum BB levels, right? And they're saying, look, here's the effect again.
yeah,
Of course, what they want to do in this experiment is then say, oh, and if we remove the cells, right, it still happens, meaning that it's nothing like it has to happen on the surface. The biochemistry has to happen on the surface of the cell in order to get that BB cleavage.
yeah, yeah. And I think that does show that there is definitely something happening. And looking at figure 4b, let's see, we've got. Yeah. So again, like the S2 and S1, they're binding to like the Fapig null cell.
Yep, absolutely. I mean, again, 4B is the same BB readout, and they're using in the presence of cells because they feel like 4A showed us that they need in the presence of cells. And yeah, and it's knocked down by the addition of that factor D, but not by the anti-C5BA because again, that's sorry, not C5BA. I saw the A from antibody, the
Yeah,
C5 antibody, because that's later on in the pathway, right? Like you can't block something later and then
yeah.
see this effect. Yeah.
So I think this I'm a bit more convinced by. That seems. I mean, again, like, the effects are much clearer here as well. Um, so that brings us to figure S6 and S3.
Yeah, S5 and S3.
S5 and S3. Sorry. Uh,
So yeah, so this is well, the first S5 is just showing us that these TF1 cells, the ones they've been using for these assays, the ones we've been looking at through flow cytometry and through lysis, they do not have ACE2
yeah,
on the surface.
yeah,
So if you stain them for ACE2, they don't have any shift in flow cytometry. Um, but they do somehow bind the S1 and the S2 fragments.
yes. And the authors like refer back to another study where they found like three binding sites on the spike proteins for heparan sulfate. So heparan sulfate is one of those. It's kind of like a sugar molecule that's found like on the surface of lots of cells in humans. So yeah, it's kind of interesting because we haven't come across that previously, and I think that's quite an interesting thing that we found through that they've
Yeah, yeah,
shown us. Yeah,
absolutely. A little insight into that. Yeah, more of the properties of the spike. I
yeah,
mean, super important. Now, I think what I liked about reading this paper, this is like a bit of a tangent, but I think it's
mm.
interesting. What I liked about reading this paper, in light of after reading all this mutation stuff, is like, tell me about all the mutations across the spike and how they influence these different phenomena. And it's going to set up because in Figure 5AB, they're using like exogenous heparan sulfate to like blast, like basically dilute out the ability for S1 or S2 to bind the cell surface, right?
Yeah,
They're saying if we just put a bunch of heparan sulfate in there, it's going to disrupt that interaction because S1 wants to bind to it, right? And then it won't be able to bind the surface. But I think an important, this is on the coattails of saying I'm really interested in mutation. A really important thing is like, oh, you just said they hypothesized that binding through three sites, right? On
hmm,
in the S1 and S2, mutate away those sites for us and show us that you can disrupt it that way. I think that'd be really cool.
that'd be really cool as well. it
And I
also,
think also like a more definite description of like the affinity, the heparin sulfate affinity. They reference the paper, but I didn't look into it. Maybe they did in that paper already.
so I think they did a paper. They referenced a paper. I looked into it, and they said that I didn't look into it much, but it said that they found three binding sites. But again, I'm not sure whether it was in. I can't remember whether it was in the whole spike protein or whether it was like out of complex, like these two versions of the spike protein workers.
Right.
Yeah. So but yeah, I think that this does kind of convince me that that's the mediator. That's how the spike protein is binding to these specific this specific cell type they're using for the MM assay.
Hmm.
Um,
By blasting it out with the heparin sulfate.
yeah, that could be the reason why it doesn't bind. I
Yeah,
mean,
yeah.
that's the
I
most
think
plausible. I can.
to me it's, yeah, it's the most plausible. It's the most plausible explanation for sure.
Yeah.
S4, they are speaking to, I think they're speaking to the factor H actually here a little bit, because they're saying like sialic acid might be like something that's mediating this interaction.
So yeah, it's weird because in their introduction they say that lots of SARS-CoV-2 viruses use sialic acid, in the
Mhm.
same way that as a binding. So like influenza uses sialic acid also as like a way of binding, and I think this is a way to like kind of show that SARS-CoV-2 isn't necessarily like that. That
Doing it.
it's that it's not binding it through sialic acid. There's not like a generic like ability of coronaviruses to bind sialic acid. There's something else going on.
Mhm.
That's how I interpreted it. I mean,
I
but
guess the other, I agree that's how they're using it, but to me it also it was part of their story, or I mean, based on how I think I was thinking about it, part of their story that like factor H is also important, like needs sialic acid to work. It was interesting they had this tool. I just feel like maybe, yeah, never mind. Maybe they did eliminate that way, and I was just thinking down another path where like they could have also used this sialidase um, in maybe their next assay.
I mean, I think you're maybe right about that because maybe I missed out on the sialic acid aspect of the factor H thing, um, but it's not quite clear like how that links into their model. If
Yeah, yeah. I don't think they made. I don't think it's explicit, but it made me think because, like I don't know the full tool set that exists in a complement lab, right? Trying
hmm,
to study the complement pathway, I don't know the best methods to go down and use them. So when I saw this and I was trying to link it back to like the very beginning where they were saying, oh, like it could also be this factor H issue, I was like, oh, it would be interesting for them to show like, yeah, without sialic acid on the surface. Um, do they still see the same reduction using the factor H inhibitor
yeah,
with factor H in? Yeah, like the same thing they do in 4A. What if they did the experimental addition of the factor H, yeah, in C and D? Anyways, that's just like a gentle experimental
yeah.
thought.
So the final thing is where they figure like C and D. So factor H we've mentioned before. It's the anti-complement thing. So it's there to regulate. So when it spots things like heparan sulfate or so glucosaminoglycans and sialic acids, which are generally present in host cells but not normally on pathogen surfaces,
Hmm.
they it binds to that. So it kind of gives like the flag like don't touch this,
Yeah,
please.
it's a bit of the it's some protection.
Yeah. Uh, so what they did was they added a bunch of factor H to the cells,
Yep,
um,
their experimental process with the lysis.
and tried to show that. And well, so I think their idea. This is where like kind of I go cross-eyed a little bit with their model because their idea is that they believe that the spike protein is causing like the alternate pathway by antagonizing factor H. Um, which would mean that if that was the case, and if you add like the S1 protein or S2 protein and you add factor H, you should still see the same amount of killing. Um, and it's uh, so
Oh
it's
yeah, you're thinking they should get full rescue of their
I mean,
phenotype.
yeah, I don't. Well, the thing is, I feel like at this point, like I was misled by the significance dots when really, like, actually, you wouldn't expect full rescue because it's not going to be perfect if you're going to flood it
Yeah,
with like factor H.
yeah,
So, but like, so if
because
you
there's going to be binding dynamics at play, right? Like how much how can you saturate those cells with enough factor H, right, that they don't get triggered in this way?
Yeah,
Also, binding affinity may play a role, right? Like binding is binding off and on all the time. Maybe when some factor H binds off, then the S1 binds on.
yeah. This is why I think like the significant dots in this particular diagram go against that model, because if you
Hmm.
take those out, then actually their model looks a lot more kind of plausible. If you ignore them completely, you're like, okay, well, it does seem like some of the complement killing activity is some of the factor H like activity is kind of still is not preventing these cells from dying. I mean, it would have been great to have a positive
Shouldn't they have
control.
just done the oh yeah. I was thinking, shouldn't they have done the significance? Oh, a positive control would be good. That's the NHS. Oh no, I guess there's no pure positive. Something else I see. Another compound, right, that activates APC on the surface of those cells. Yeah.
Yeah, and then you'd be able to see like how much that because
Yeah. Oh, and then you add factor H into that one too, maybe,
yeah,
right? And you would see that. Yeah. So actually, maybe they should have used the AHUS serum. It would be nice, right? That's actually where this assay is, right?
yeah.
If they're making the argument that it's because of what we've seen, right, that it's so close to this particular pathogenic process, maybe AHUS serum here plus factor H would give us the bar to compare against.
But at the moment, we don't have that comparison because what we'd either say like, oh, it abrogates all activity, but then if we're saying that, then we're also being assuming that S1 would do. So I guess it's kind of like almost like an orphan data point that's there that we need further context around in order to figure out what it actually means.
Yeah. I mean, they show us like a small increase, right? Like, oh yeah, I don't know what to say. Yeah,
Yeah,
because
it's
brings
because
it down by a significant amount, but is it more than NHS?
yeah, this is
NHS should also have factor H in it, I guess, to also be really tight. I'm pretty sure though it's probably taken for granted that when you add factor H into NHS, it doesn't produce any extra
well,
deposition.
yeah, because again, unless you've got something to trigger the complement, it won't. There won't be any So there's the inhibitor won't do anything. So if you need to have something that triggers the complement, and then we have factor H, so you can see exactly how much factor H can neutralize complement activity in like kind of something that we know activates complement. Whereas in
Hmm.
here, we don't necessarily know whether like it could be that the factor H isn't very good at blocking things, or
Hmm.
and this is like how normally it would look like if it's blocked, or if I mean it's and when you get down to it, you've got something that inhibits complement, and we find that complement is slightly inhibited, and it's not necessarily like what you'd kind of expect that. So it's we
Yeah,
need to
but they reconstituted the trigger of the complements, right? Like as S1 and S2.
yeah. Yeah, I mean, I guess you could try and get. I mean, if they compared them side by side, I guess you could go by looking at the differences of them, or even if you could do like a kind of a dilution curve and then measure like kind of the rate differences. Perhaps you'd
Hmm.
find something. But yeah, this doesn't
Yeah,
really. Yeah, it it doesn't
because
fill me with confidence,
I,
but
yeah, I mean it's interesting. So if you flip to E and F, right? In E and F, the addition of factor H is more similar, right? It's to
yeah,
knocking down to the baseline level of just NHS.
which actually goes against their model because then it's you're basically saying, okay, well then spike protein doesn't interfere with the interaction between factor H and the heparan
Oh,
sulfate.
sure,
So
sure, sure.
that
It can't
actually,
kick it off. Yeah, it can't kick
so
it.
so yeah, that actually goes against the model that they're proposing, which is really
Well, I
unfortunate.
mean, yeah, yeah. Well, I mean, I guess that because the two segments are different, right?
Mm.
Right, and of course, inside of where we would expect to see this phenomena is in the blood with actual viral particles with full spike protein, right? And they've reduced us down to just this like biochemical in vitro model system. But yeah, you're right. In terms of strictly interpreting it, this piece is like, oh well, I guess S2 is not the important one, is what it's trying to
Yeah.
say.
So it's
But
basically saying, but then that doesn't quite sink in with what we've
but
seen
that
with
doesn't
the.
sink in with what we've seen with lysis and earlier, right? With the
Yeah.
deposition using S2. Yeah.
Unless you go by the idea that none of this matters because nothing's like got enough power to it, so you're gonna it's all
I mean,
random. But
I would
let's not
think
be
that
that nihilistic.
the reason why that they feel confidence in what they're seeing is that they like because remember these readouts are also very artificial, right?
Mm.
These readouts are kind of like chosen um they're in vitro readouts, first of all, right? They don't have biological relevance, right?
Mm.
They're assays made for this scenario to show us whether or not like these purified components will do what we're hypothesizing they do. And it does come out that S1 has some sort of effect. Yeah, I agree. The statistics are weird. I wouldn't know how to do this one better because you want it. Yeah, maybe
Uh,
the dose, maybe the doses would help if you add more as you add more FH, right? You
mm.
get more inhibition. That would be great.
Yeah,
And
if you yeah.
Nova that, right? Don't do this t-test thing.
Yeah, because
Yeah.
I feel like there could be something here, but I need, but again, I don't feel confident about it, and I would need a lot more data to get that firmity, and I don't feel like this paper's give me that security yet. But it's,
Yeah. But that's also speaking as a not a complement person,
yeah,
right? Not a person in that field who may be like, yeah, we see stuff like that all the time, and we say, right, like if someone showed you some historical papers, right, where they proved certain principles and this data was like comparably rigorous, right, you'd be like, oh, actually, I feel pretty confident. The statistics were still bad, right? They still could have done much better, right, to convince me through their data. But like we've seen phenomena like this in the past. That could help, right? Confidence boost you probably
yeah,
into
I
belief.
think, yeah, I mean, I feel bad that saying like, oh yeah, the thing that confidence boosts me is that there are other papers out there, which is like the plenty of fish in the sea kind of argument of,
No,
eh,
but I guess what I'm saying is norms in a field, right? That
yeah,
plays into the way people interpret things, right? Like we come into reading this with no norms, right? And so
yeah,
from the strictest lens, right, of our own statistical understanding and how we've understood the system, we're not entirely convinced, right, that the model that they present at the end is good. There's like critical pieces of data maybe that are missing.
yeah,
The data does suggest though the model, right? Like
mm,
the way they put it together, like yes, this could be happening, right? Maybe we'd want to see some follow-up experiments or just these same experiments done in a slightly different way, presented in a slightly different way.
yeah. I feel like yeah, I feel a lot like that. I feel like this didn't really do it for me as a paper in terms, but then we have been spoiling ourselves on like Nature papers and other like kind of papers with which, and this is a much more like kind of yeah. Again, I don't want to say like kind of the complement field isn't like this is so yeah. I don't want to say anything negative about this paper because I feel like there was definitely effort put in and that they definitely tried to investigate a question. They came up with some answers, but the actual certainty I have with those answers isn't necessarily
I
very
guess
high.
for me, when I finish reading a paper and there's some doubt cast on it, to me, I just want to know what did we learn, right?
Yeah.
like I just want to know what did we learn after reading it because like it's sometimes impossible to create the most um, airtight arguments using data, right, for your
Mm.
hypothesis, right? Like that's not. I don't think that's the purpose of papers
Yeah.
either, right? To
Yeah.
be
You're
a
right.
single airtight argument against or for a certain hypothesis. It's instead to show us like sort of the way in which you would go to show, right? Like it's key pieces of information that are being
Mm.
shown, and we're trying to see does that align with the way that they're thinking. Yes, the data that they have does align with their final model, right?
Mm.
But we
Mm.
can't accept the full final model because some of those pieces seem shakier, right, than we would think. But that's why I'm trying to push us to say what did we learn because right now
Yeah.
we've seen a lot of uncertainty.
Yeah, okay. So I guess we've learned that SARS does interact with the spike protein. There is some interaction with complement there. I feel like I can say that, and perhaps there is some interaction with heparan sulfate.
I agree. I'm not so sold on the heparin sulfate interaction because they just blast out with heparin. I mean, that's like people's
Mm,
interpretation, right? Do you believe that this adding exogenous heparin sulfate is the best way, right, to show just in one way, right,
mm.
that interaction exists? And then that's not is that lysis? No, that's just binding of S1 and S2, right? Like to me, that's not even the most sensitive um, that's not the most sensitive output of this. maybe seeing the lysis information would have made me feel better, or just seeing it in addition. I don't know now. Now I haven't thought so much about like how I would change some of these to make it more convincing to me, and I'm not going to be able to do it as we're
Oh no,
streaming.
we're not getting able to like create an entire new grant application while we're streaming. I mean,
Yeah.
that's asking a lot of
Well, no,
ourselves.
like I just
And
feel like from what I know now of this experiment, how would I take the tools that they had and made it better? To me, that's like ideally that's what reviewers could help out with. But of course, having reviewers that aren't in your field, like us,
yeah, yeah, it's not gonna be.
isn't really we're not going to be the source of those important next
Yeah,
steps.
I mean, I say, oh yeah, we could do a dilution series. I don't know how much human serum they have on them. I mean, they could be like, because if it's like anything like my lab, it comes from the people who are working in the lab. So I mean, it's like, so that is a limited resource. So if, I mean, I don't want to like exsanguinate one of the researchers to just because I'm like, oh, I have comments. I mean, sometimes this is like the best we can get with what the resources we have
I
for this
did
moment.
like the experiment though, like just for figure 5 CD, right? To better get at like what is this reduction? This
Mm,
not full reduction but partial reduction with the addition of FH. If they titrated that and showed it was dose dependent, then we could really like map it back to FH, I guess.
yeah.
Uh,
Yeah, I feel like that kind of biochemistry would have been really helpful, but yeah, again, we don't know what resources they had to and in SARS-CoV-2 there is that time that people have
yeah.
to think about.
Yeah. Oh, sorry. I just like I we're in the zone of like thinking about other experiments, and it just made me think. What about competition of S1 binding, right,
Ooh,
between heparin sulfate and I don't know, like because it's not that the HS binding proteins are binding. What are they binding here? Like, do they recruit factor H? That's why I don't know. What's the relationship between factor H and the heparin sulfate binding proteins? If I knew
so
that relationship better,
yeah,
I could imagine maybe a competition experiment of some sort, right? Competition of binding could give me like a better like, yeah, this phenomena is happening.
yeah, and in terms of like the wider thing, I mean, complement is so complex. I think this highlights an important thing that we wouldn't, because usually I'd assume that the normal pathway would be the main thing that was taking effect during sepsis. That would be my usual assumption because you've got antibodies binding to binding to creating complexes. So that'd be what my assumption of like, oh, if I see endothelial damage due to complement, that's what I'm assuming is happening. But this
Yeah,
kind of sheds light onto a different aspect of the complement pathway that I wouldn't
that's
have necessarily thought of. So I think that's something that we can take away.
yeah, no, that's a good. I like that because that actually that's about we have a paper in our Zotero, right, that talks about thrombotic autoantibodies, right? And
Yeah,
there's a line of thinking saying that these phenomena, this clotting phenomena, might be this type of sequelae action with like widespread activation of the immune system means autoantibodies are being made, right? Which then leads to the clotting. That's not what this is proposing, right? This is
no,
proposing on the alternative pathway. And actually, both could be happening.
yeah, that's oh yeah, that's the tough thing.
This, right? Both could be happening. And like, we haven't been able to sort out those things. But yeah, no, I think that that is a very good thing to draw from this, right? That the alternative pathway could be, right, one of the ways in which SARS-CoV-2 creates these strange clotting symptoms.
Yeah, I'm willing to go with that. I think that I want to leave it on a high note because mining because we want to be positive about papers. We like reading papers. We like analyzing science, and yeah, sometimes we want more. And I feel that's a lot where like anything that seems critical is coming from is we want more information, and sometimes you don't
Yeah.
always get that from a paper.
Yeah.
And I'm just sitting here with my blue balls, thinking I need more data, and
Yeah, and I think it's important that I mean you've said it already many times, right? Like authors themselves are also under limitations, right?
yeah,
For resources, for time, for space, right? Like to get published for their own careers. Yeah. So the fact that we are able to learn anything from a paper is like that is always a positive, right? That something came out of it. Um,
yeah, and we didn't really. This is the first time we've really looked into the complement in all of our like kind of. In I think it's since like May we've been going. We haven't looked at it yet, and I think this was a
yeah.
good chance to like brush up on what's going on with complement.
Yeah, I think I saw like as I was reading this, as I was reading for background on this, I saw something about like neutrophil activation. I remember we have a preprint about neutrophil activation,
Yeah.
and like I forget, yeah, you forget that there are these other proteins, this other innate immune system support network, right? That's acting on that can lead to pathogenesis, but also that could be protecting against SARS-CoV-2 in a certain way. And how do you nudge those systems, right, to be therapeutically relevant? And or how do you look at what's happening and say, oh, this is, like this is a cool function, right, of viral proteins, right? Like here are epitopes that can be used to tickle the immune system in different ways. Like you don't know where that research will go. That is always exciting. Okay. Well, join us next week for our news week where we will survey papers to find something to cover like this in the following week.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections you want to give us, then please let us know in the comments.
Yep,
Um,
I wholeheartedly agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that this is a peer review process that we're doing, and we hope that more people can participate with us.
yeah. So it's been a pleasure talking with you, Danny.
Same here.
Yeah. So catch us next week. We'll be our news show. So we'll have much more microbiology content.